hi everyone. My name is Michael. I just finished my second year in Section Jun's group. I've been working in biology at all for just over a year. So forgive the rather generic title. I'm interested in exploring like the division and transformation mechanisms in the minimal cells, and I'll talk a little bit about how I'm going to do that and why. So first, a little bit about our group. Broadly speaking, we're like a biophysics group, and we study bacterial physiology. So how E. coli divide, how replication, the MIN system, that sort of thing. Broadly speaking, our hammer is the mother machine. This is a microfluidic device with about micrometer wide channels with a trench that carries nutrients. It's essentially a chemostat. So here is a video of some E. coli growing inside of this mother machine, and basically there's a strong flow going over the top, and as the E. coli leave these channels, they get swept away by the flow. We also have done some kind of work with basically microscopic pistons, and actually one of the people here, James Poitier, I think, was really involved in this work. And essentially what you find is that under confinement, chromosomes behave like springs. And I'll explain why this is relevant to the minimal cells in a bit. All right, so so last year, I'd only been working with the minimal cells for a little bit. So what I'd had a chance to do is grow them in these microfluidic devices. Here we have JCVI Syn3O, which, as you may or may not know, has this extremely pleomorphic phenotype. Essentially, this is what happens with mycoplasma when you disable its kind of division mechanisms. It filaments, but unlike E. coli, for which filamentation is basically lethal, this cell doesn't mind so much. In practice, the doubling time goes from 2 hours to 3 hours, and you get all sorts of really fun shapes. You get these cool tentacles. You get interesting sort of spontaneous kind of vesicle formation. These don't contain any cytoplasm. And that's just sort of here on the right. It's just kind of an image of what all that kind of looks like when it's in a large channel. Syn3A and the other kind of more typical morphology synthetic minimal cells, they're kind of growing in the mother machine. I haven't had as much time to work on this project as I would have liked, but I'll talk a little bit about kind of my future plans with these cells. So goal number one is to explore kind of the division of these organisms. Now, why is this interesting? Well, part of the reason is that it happens to be a pretty easy to work with mycoplasma, and mycoplasma in and of itself is also a very interesting kind of variety of organism. There's a strain of Syn3O called Syn3O plus 126 that contains seven genes that are essential for division. Among them is FTSZ and SEPF. And basically, if you knock down any one of these genes, what you find is that the genes exhibit this kind of really fun pleomorphic phenotype that we see in Syn3O. With those 7 genes, we find that it looks like the more typical kind of mycoplasma JCVI Syn3A sort of situation. There's a really fun story sort of shaping up already with FTSZ. For starters, in the genomic context of Syn3O plus 126, knocking down FTSZ causes this pleomorphic phenotype. But in the context of JCVI Syn3A, which contains a few other kind of extraneous genes, what you find is that it does not cause this pleomorphic phenotype. Sort of to thicken the story a little bit, the recent Nature paper about minimal cell evolution, one of the kind of salient points was that there is a mutation in FTSZ that, when introduced into JCVI Syn3A, makes the organism slightly smaller and increases the growth rate. But when that same mutation is introduced into Syn 1.0, what you find is that the cell actually gets a little bit bigger and also accelerates in growth rate. So what I'm kind of hoping to do is to do a knockdown of these 7 genes in the Syn3O plus 126, using a CRISPR-I system. This way, what we can do is not only have cells with the genes on and off, but we can actually do a kind of a gradual knockdown. And hopefully, by doing this, we'll kind of be able to see a transition between these kind of regular mycoplasma morphologies and the more pleomorphic phenotype. There's kind of a lot of work I'll probably need to do. It's very hard to gauge the size of Syn3A kind of in the chemostats that we use. So I think we're probably going to be trying to set up sort of a super resolution system. But yeah, that's sort of the general plan in kind of the near future. Another sort of stretch goal is, I think, really fun, and it sort of actually derives from a lot of follows a lot of work that James Pelletier again was doing. Is we'd really like to be able to see genome transplantation happen like in person, let's say. So genome transplantation is unlikely, but it's not that unlikely. If you look at the SIs on the 2016 paper, what you find is that there's about 10 million cells in a sort of typical genome transplantation experiment. With 500 to 1,500 transplants, that means you need that means you really don't need that many kind of cells before you see a transplantation. Now, just coincidentally, our lab's been doing some extremely high throughput imaging where we have 2,000 kind of these channels, and we have 16 sort of inlets. So you're able to do just gigantic numbers of sort of growing cells at the same time. So sort of the plan there is we're going to try and follow, just start off with some regular transformation protocols on kind of shorter plasmids, and depending on how those go, if we see any kind of anything interesting, we might try and scale up to trying to do a whole genome transplantation in one of our microfluidic devices. Right. So honestly, that's it. Like I said, I haven't had a chance to do as much work as I would have liked in the last year, but I'm hoping next year I can present some interesting results. So I wanted to give a quick thanks to, of course, John Glass for getting us started, Sage Glass for a lot of help with kind of just basic minimal cell stuff, James Pelletier for some great guiding conversations, and of course my PI, Section June, and the rest of the lab. All right. Yeah. So I'm happy to take any questions.
Michael, you may need to stop sharing and we'll get reverb.
Oh, I'm wearing earbuds.
Right. Questions, anyone?
I was trying to think of a good one related to the fact or some of the previous data that's been generated in with the FTS they found.
Sorry, can you say that again? You're cutting out.
Oh, sorry. I said I was trying to think of a good word for this, so I apologize. I know like some data out in the literature that FTS and Italium are tolerated, and I know it's obviously in tandem with it being in Syn3A, like denotated as like a quasi-essential gene. Yeah, sorry.
So yeah, so I kind of know a little bit of what you're talking about. So yeah, basically it seems that because mycoplasma don't have a cell wall, they generally tolerate filamentation. There's a really cool paper by Jeff Arrington where he's sort of playing with L-forms and doing a lot of very similar experiments in very kind of similar kind of microfluidic chemostats, but he's doing them with L-forms from B. subtilis. And what you find is that, broadly speaking, when you have these sort of wall-less bacteria, they maybe it's due to shear forces. Maybe it's due to kind of spontaneous curvature. There's maybe it's probably honestly due to both. They tend to be capable of division, and I think that there's some kind of suspicion that this is probably some like pretty primitive division mechanism that could have been present in early life. So yeah, FTSE, but also. I don't remember if this is the case in genitalium, but I know that the other kind of like even Syn3A, if you remove FTSE, it divides just fine. Like the phenotype is mostly preserved, and I'm kind of hoping that once I get a sort of ISIM imaging setup, I can actually check if there's any subtle changes in size. But right now, just because of like the diffraction limit, it's quite hard to get a gauge of how big exactly the Syn3A sizes are without kind of doing fixation and looking at them in an electron microscope. Yeah.
Well, thank you. This is a really interesting stuff.
Yeah, I'm really excited about the kind of graduate knockdown experiments. It's gonna be really cool.